Okay, I think we can start. It's 3:04, 3:02 actually. So good afternoon, everyone. I'm Cinzia Perrino from Federico II University in Naples, Italy. And this is, I think, a very interesting and important webinar on exosomes that the working group of cellular and molecular biology of the heart of the Italian Society of Cardiology organized. And we have the honor of having today one, I think, of the most important experts on this field in cardiology and particularly in cardioprotection. There is Sean Davidson that is already here with us. Now, Sean is a friend and also is a brilliant scientist. He got his PhD. Actually, I didn't know that, Sean. I learned investigating your CV. Actually, he got his PhD in Australia, in Melbourne. I didn't know that.
Oh, that's where I'm from. Yeah. I
Yeah.
was born there. Yeah.
That's interesting. And then he did his postdoc in Paris, which is also very interesting. And since 1998, he is in London and basically is a professor at the After Cardiovascular Institute at the University College of London. He is past chair of the ESC working group of cellular biology of the heart. And as I said, he's an expert on exosomes, and not only exosomes, but is one of the major focuses of his research and cardioprotection. So thank you, Sean, for accepting the invitation. And I asked Professor Massimiliano Ruscica from University of Milan to give a very short introduction on the topic, and then we'll have the discussion. Thank you very much.
Thank you, Cinzia, for inviting me to co-chair this session, and thanks, Sean, for this opportunity. Really, I'm very honored. And forgive me, but I used your recent review published on cardiovascular research to give an overview on this important topic in which you described the importance to understand some drawbacks when we talk about exosomes and microvesicles. So sometimes there is confusion, and most important is to understand which are the techniques and the way to define the correct interpretation, so the correct isolation of exosomes and microvesicles, since these particles, the nanoparticles, are promising also as a therapeutic agents. So I'm sure that being extracellular vesicles in general important in all the processes of atherosclerotic burden, I'm really sure that today we'll learn more from your presentation. Thanks, John, and please, the stage is yours.
Thanks very much. And thanks, Cinzia and Massimiliano, for this lovely introduction. And thanks for inviting me to speak here today. I'll share my slides. And you should see those now. So Cinzia kindly asked me to speak a bit about exosomes and some background about what they are. So I understand there's a mixed and translational clinical audience. So we'll try and give you some background about what these vesicles are and how they can benefit patients with cardiovascular disease. I probably don't need to show this slide, but actually I like to introduce it just to put the context of our interests is in cardiovascular disease, and in particular in myocardial infarction. And it's also a very dramatic slide, so I think it's a good one to start with. And so what we're interested in, I'm sure you know about ischemia, when we have myocardial infarction and ischemia occurring in the heart, and that as therapy, reperfusion can limit the extent of infarction. But what we're particularly interested in is trying to target not just ischemia, but reperfusion injury, this injury which can occur due to reperfusion. So although this is necessary, reperfusion itself can cause further infarct. And we know that at least experimentally, because we can intervene at this point of reperfusion and reduce the extent of infarct. Looking at this is sections of an animal heart taken after ischemia and reperfusion with the infarct in white. And with intervention at reperfusion, we can limit the extent of infarct. Obviously, infarction is bad not just for the fact that it causes infarct itself and damage to the myocardium, but can result in a greatly increased chance of heart failure down the line. And you'll be aware also of the major influence of diabetes in the risk of MI and the worsening effect of diabetes as well. So these are all themes I'll come back to in relation to exosomes as well. What I'd like to cover today is a bit of background about what exosomes are, for those who have not come across them so much before. And as Mesenchymal mentioned, there's some important technical aspects of how to actually work with exosomes. It's not so easy to work with these nano-sized particles. And then a bit about how we can actually use them potentially to protect the heart from MI in different settings and in the settings of diabetes as well. So exosomes, as you heard, they're a type of nanovesicle, and there are a number of different types of lipid-bound vesicles which are released from cells. As you can see here, these are 3 examples, microvesicles, which tend to be slightly larger and shed off the surface of cells. Exosomes is what we're really interested in, and these are produced by invagination of these membranes of these endosome bodies. And once these lipid vesicles form inside these multivesicular bodies, when these fuse with the outer membrane, they can release the particles or the vesicles as exosomes. And we could recognize them by the specific proteins, markers expressed on the surface of the exosomes. And there's been a lot of interest in these particles in the last 5-8 years or so. And these aren't the only type of extracellular vesicle. There are also cell vesicles released from dying cells in the form of apoptotic bodies, for example. And since when we isolate these and try and work with them, it's very difficult to obtain a completely pure population. So more strictly speaking, we should refer to them as extracellular vesicles or EVs when we're using the product. But what we're particularly interested in, as I mentioned, is these exosomes, because they contain proteins of origin from the cell, so can be used as potential biomarkers from the cell of origin, but also have signaling roles between cells, which appear to be important. So where do they come from? They are produced by just about every cell type, it seems. All cells that have been studied release these exosomes, or small EVs. And they're found in the blood, but there's been particular interest in those which are produced from stem cells. So for example, in cardiovascular research, stem cells studied for many years as a way of trying to potentially regenerate the heart. But now interest has changed slightly towards these EVs produced from stem cells, which appear to mediate a lot of the benefit which has been seen from them. So they signal to the heart and can have multiple different beneficial effects in modulating the immune system, in stimulating angiogenesis, which is obviously beneficial, in reducing fibrosis, and in protecting the heart from infarction as well. And you'll notice on here that there's plasma itself. Blood plasma is a source of exosomes or EVs. There are many of them found just floating around in the blood, if you like. The mechanism by which these EVs have effects on cells can be via different mechanisms. They have proteins on the outside of the membrane, and so therefore can potentially signal via receptors in recipient cells. And they contain proteins and microRNAs within the membrane of the EVs, and so can deliver the contents to recipient cells as well and have effects on the cells that way. So because they have these multiple different mechanisms of acting, we like to think of them as a sort of a multi-tool with multiple effects, multiple effectors on them. Although it's worth remembering that they really are nano-sized vesicles, smaller than the wavelength of light, around 50 to 100 nanometers. So if you can imagine trying to work with these vesicles, which are actually smaller than wavelength of light, you can't directly visualize them with light. We have to use other techniques. So they're really tall, but are really small, but in aggregate can have effects on recipient cells. So how do we actually isolate these EVs or exosomes? I'll mention a few points, but I'll just take this opportunity to highlight this recent review paper, which we published, speaking exactly about these methods for identifying and characterizing extracellular vesicles in cardiovascular studies, because these technical approaches, while they might sound like dry type of issues, they're very important, really crucial for understanding what it is we're actually dealing with and how they work. I'll just highlight one figure from this review, which illustrates the important aspects about when we've isolated EVs, how do we know what they are and how do we know what we're using and how pure they are? And these are 4 important steps. One is just to quantify the amount of EVs that we have relative to the starting material. So if we started with 2 mils of blood, how many EVs do we have? And there are various techniques which can be used for that. We need to show that there are actually these exosome protein markers that I mentioned present there on the EVs. So we can do Western blot or ELISA to show their presence. And that's indicative of there being exosomes present. Ideally, we should exclude or show the absence of markers, proteins, which should not be present in EVs as an indication of their purity. So for example, in blood, there's huge quantities of albumin and lipoproteins, which can be easily mixed up and difficult to separate from pure EVs. So ideally, we'd show that these are not present in a good isolation of EVs. And we use electron microscopy to look at individual exosomes. As you can see here, the size, as I mentioned, really tiny, 100 nanometers across. And we have these kind of collapsed ball appearance of these EVs using this technique. But that way, at least we can prove that they're really present. Sometimes it's nice to actually visualize and see that we have something there in our field, and it's we do have something that we're working with. But to illustrate this problem or challenge in isolating from blood, I've shown here a comparison of the size of particles and the density of particles. Here we have exosomes and microvesicles, these tiny particles, microvesicles slightly larger, but they're in a similar size range as many of the lipoprotein particles, and even a similar density to some of them, which makes it difficult to isolate. But what makes this really difficult is the quantity of these EVs in the blood has been estimated around 10 to the 8 per mil. Sounds quite a large quantity until you realize that there are actually around 10 to the 16 lipoprotein particles per mil in blood. So a massive concentration of lipoprotein particles, 8 orders of magnitude or so more than the EVs that we're interested in. So trying to purify out these small vesicles away from the bulk protein which is present and lipoproteins is actually really challenging. And this, it might be a little technical, this slide for the audience. I hope not too technical, because I would just want to illustrate some of the important aspects about why it is important to get these techniques right. This is from a publication of a PhD student here a few years ago, where we wanted to compare what is the best technique to try and isolate these small EVs from the blood. And we compared 2 popular techniques. One is being called the gold standard technique in the literature of differential ultracentrifugation, using an ultracentrifuge to pellet down their small vesicles. And another technique, which is using a size exclusion chromatography, basically a column where we can put the sample on top and separate out things by size. And interestingly, we found a massive difference just using the SEC technique. We found a very high yield of numbers of particles compared to the ultracentrifugation. So that seemed an attractive way of isolating the EVs. But this is just looking at the numbers of particles, small particles. When we actually looked at what these particles might be, and we looked for the presence of this marker protein, we found a much higher quantity of this marker protein in the ultracentrifuge samples. So the reason for that, looking now at the electron microscopy to see what's actually in the sample, we can see that the SEC purified sample, we have lots of these lipoprotein contaminants, which are just spherical, these L indicated by L here. Whereas the ultracentrifuge sample, which had more of the exosome marker proteins present, actually have more of these nice EV exosome type appearing vesicles. So in the end, actually the ultracentrifugation method is better, And to confirm that, we can show that the presence of the apolipoprotein B, this is a protein associated with lipoproteins. There's lots of this in the plasma, we know, but in the SEC purified technique, but not encouragingly in the ultracentrifuge sample. So this makes sense actually because the SEC technique is separating mainly by size, and lipoproteins, we saw they're a similar size to the smaller EVs. So anyway, this is some preliminary experiments, if you like, which confirmed that the ultracentrifuge technique is good for isolating more pure EVs away from the contaminating lipoproteins. And we went on to use this technique. We isolated EVs from the blood of either rats or humans, and you can see they have a similar appearance. They both contain these marker proteins for EVs that we're working with. And we used nanoparticle tracking analysis, it is, but it's looking at the size distribution of these particles. We could see they're all around this nano size, less than 200 nanometers in diameter. So really these tiny nanoparticles. And our interest back then, a number of years ago, was, are these small EVs or exosomes which are found in the blood, do they have beneficial effects in the cardiovascular system? And in our lab at the Hatter Cardiovascular Institute, we're interested in particular about cardioprotection from ischemic reperfusion and reduction of infarct size, as I mentioned. So we isolated EVs from the blood of rats, as you can see, and in addition, what we were particularly interested in, our hypothesis really, was whether they might mediate a type of cardioprotection which is called remote ischemic conditioning. And you may or may not have come across this. It's quite an interesting phenomenon which has been shown reproducibly in many different animal studies, at least, and there's evidence for it working in humans as well. And this is where a short period of ischemia is actually applied deliberately to the arm. So if you make the arm ischemic for 5 minutes, and here it's shown using a blood pressure cuff, but this is inflated to very high pressure, suprasystolic. So we actually get ischemia in the arm. And then when we reperfuse this can signal to the heart and induce protective signaling pathways in the heart, which will protect it from a major ischemia reperfusion episode. So this phenomenon has been known for many years, but the mechanism by how it works had not been known. And I heard about these exosomes which had been discovered not long ago and hypothesized that these might mediate the signaling from the limb to the heart. It seemed like a nice hypothesis, but hypotheses don't always go according to plan when we do the experiments. Our first step was to actually take some human or volunteers, perform the remote ischemic conditioning on the upper limb as shown here. And then we drew blood from the arm and measured the numbers of particles or exosomes in the blood. And here you can see actually there was a significant increase in the numbers of particles in the blood of those individuals who had received the RIC treatment. So this first initial part of data does support the hypothesis that this RIC remote conditioning might be releasing exosomes or smaller EVs, which are signaling to the heart and protecting the heart. So far, so good. It's not a huge increase, but it's a significant increase. So what about if we actually take these small EVs or exosomes? Are they protective? And to investigate this, we use a small animal model, a rat model in this case, where an anesthetized rat is intubated, and we open the chest, expose the heart, tie off the coronary artery for 30 minutes, and then reperfuse for a further 2 hours. And at the end of the experiment, we can take out the heart and we can stain the live tissue red, as you can see here. We can see the region of infarct and we can measure the size of that infarct. And we can see the total area, which is ischemic around here. The blue indicates the non-ischemic area. So we're interested in the infarct area relative to the total ischemic area. And if we repeat this experiment in a number of rats, in 6 animals per group, I believe it was. We can measure the infarct size in those rats which were administered just the vehicle and then subject to ischemia and reperfusion. And we have nearly 50% of the ischemic area is infarcted. In those rats, some rats, we applied this remote ischemic conditioning protocol directly to the limb. And we do this using basically a tiny pressure cuff on the rat hind limb. We can occlude the blood flow, reperfuse it, and as I said before, we can reduce the extent of the injury very significantly, more than a 50% reduction in infarct size. So we took exosomes which had been isolated either from a control naive rat or a rat which had been treated with the remote conditioning, and then injected them into this rat prior to the ischemia and reperfusion, and then measuring the infarct size. And what we can see, we weren't expecting this, but we saw either those from control rat or conditioned rat were protective. So this was somewhat surprising, but we realized that when we're injecting exosomes, even from the control rat, this is doubling the numbers of exosomes and might be sufficient to induce a cardioprotective signaling response. But the exosomes itself from the rat blood appear to be protective in a second rat. We did further experiments. Now we can take out the heart from a rat and perfuse it for up to several hours using a Langendorf perfused heart technique. This is an old technique, been around for over 100 years, but as you can see, the heart is beating. We can tie off the coronary artery, and then we can perform a similar experiment, measuring the size of infarct at the end of the experiment. And we can perfuse the heart with exosomes from either a control or conditioned rat. And in both cases, you can see this reduces the size of the infarct compared to those hearts which received just a vehicle. So the exosomes are acting directly on the heart and able to induce a protective signaling and reduce infarct size. Finally, we can actually take out the cardiac myocytes from the heart, as we've done here, from the adult heart, these rod-shaped myocytes in the dish. We can subject them to hypoxia and reoxygenation period to mimic this ischemia reperfusion in vitro. And we can administer, here we've used an increasing concentration of exosomes either from control or conditioned rats. And you can see a nice dose response. The more exosomes we put in the dish, the more resistant they are against the hypoxia and reoxygenation. Interestingly, we do see a slightly increased protection consistently with those from the conditioned rats, although it was not significant. So what was significant was the fact that the exosomes were protective. I don't want to go into too much about the molecular details, but we looked a bit about the mechanism of how they might be protecting the heart as well. And to do this, we did Western blots of the cells which had been treated with exosomes for increasing or a range of times, 0 to 30 minutes. And then we probed for phosphorylation or activation of this kinase, ERK kinase. We saw increase in phosphorylation after 5 minutes of treatment. No response in terms of the AKT signaling pathway, which is, we know it to be highly cardioprotective, but in this case, the exosomes activated only the ERK pathway. The right-hand side is a positive control. And then to see whether this kinase pathway was important in the mechanism of protection, we used 2 different inhibitors of the kinase pathway. These are called UO126 or PD98059, 2 chemical inhibitors which prevent ERK activation. And here we've used the cell experiments again. We have around about 20% cell death in normoxic healthy cells. Those which are treated with hypoxia and reoxygenation, we see a significant increase in cell death. The exosomes, as we saw before, they reduce the amount of cell death. But in the presence of the inhibitors, we lose that protection. So that ERK MAP kinase signaling pathway is an essential part of the protection mediated by the exosomes. And I won't show all the details, but we used a series of different inhibitors to elucidate this pathway where it's actually the heat shock protein HSP70 molecule, which is on the outside of the exosomes, can act as a signaling molecule to stimulate TLR4. This is called a toll-like receptor 4. part of the innate immune system, which is known to lead to different signaling pathways inside the cell and activate this signaling pathway. So we believe we know how these exosomes are protecting, and that if we increase the quantity of these exosomes, they can activate these signaling pathways. Just to show you a bit more about where these exosomes actually come from in the blood, we've used a different technique. This is some equipment which allows us to use antibody markers to see where these exosomes are actually coming from. So we can see just in healthy blood, this is healthy human blood, we can see, in fact, most of these small vesicles, the exosomes originating from platelets. There's some contribution from the endothelial cells as well. So as I mentioned, there are a large quantity of exosomes in the blood endogenously, and many of these coming from platelets, also from endothelial cells. This is just the output of that study, which was interesting because it also can tell us the size range of the particles, and you can see the endothelial derived ones around the exosome sized range of 50 to 100 nanometers primarily. The platelet derived exosomes are a slightly larger range, which we believe might be more consisting of the microvesicles. So we believe the endothelial exosomes may be the protective ones. I'll show you a bit about the effect of diabetes, because as I mentioned, the diabetes we know can have a strong influence on the protection from these exosomes. On the protective or an influence on myocardial infarction. We wanted to know whether diabetes would affect protection from the exosomes. And we did this study mainly using a diabetic rat model, shown here, type 2 diabetes rat model, although some of the data was also replicated in human diabetic and non-diabetic patients as well. In both samples, we showed that there was no effect of diabetes on the appearance of the exosomes or the diameter of the exosomes or the concentration in the blood. I'm showing you here the data from the rats, but similarly, there was no effect of diabetes on the exosomes themselves in human blood. However, when we looked at whether the exosomes were protective, we treated the myocytes in vitro. We subject them to hypoxia and reoxygenation, and we see cell death here. The non-diabetic exosomes were protective, but those from the diabetic rats here were not protective. So the diabetes appears to impair protection. And looking at the signaling pathway, here again, we could see that the exosomes from the diabetic rats were no longer able to activate this signaling pathway. So this signaling seems to be impaired in diabetes. Importantly, though, if we were going to use exosomes as a protective treatment for patients potentially, we would not want to know whether non-diabetic exosomes would protect in the setting of diabetes. So we did an experiment where we have myocytes from a diabetic rat treated with exosomes from a non-diabetic rat. And here we can see that when we isolate the exosomes from a non-diabetic rat, they do reduce hypoxia or reoxygenation injury. So the final part of the talk, I'm not going to go through all of this because I think it might take too long, but just to cover a little bit of what we're actually interested in more as a type of therapy is looking at exosomes from mesenchymal stromal cells, also sometimes called mesenchymal stem cells, MSC. These are an interesting type of stem cell or stromal cell because they can differentiate into multiple cells and also reduce these different factors which can have effects on distal cell types. And we were interested in particular about the exosomes that they produce. In our studies, we've looked at those MSC cells, a specific type of MSC, which are isolated from the amniotic fluid for various reasons, but these have high levels of pluripotency markers and can be expanded well in culture and seem to have interesting characteristics. So we've cultured these cells in vitro. We've isolated them here using size exclusion chromatography, which when the cells are cultured in serum-free conditions allows us to get very pure EVs. And I'll just show you to illustrate the purity here. We have the small EVs, the exosomes isolated coming off early on the column, the protein, the bulk of the protein comes off much later. So we get very good separation and very pure exosomes. We can see that also from the exosomal marker proteins coming off early in the column. We characterized them to show that the small exosomes here have the appearance of exosomes that we expect. They range from 50 to 200 nanometers primarily. And the important experiment was here we've used actually a mouse model, but we've injected them just prior to reperfusion intravenously. And in the blinded analysis, this is a blinded study where we didn't, the tubes were not labeled, unlabeled, coded basically. We've administered either vehicle or the small EVs from the MSC cells, or a positive control of bradykinin, which is known to be protective in this model. And as you can see, we saw a significant reduction in the infarct size with either of these two treatments. But the small EVs or the exosomes were very protective in this model with less infarct visible. I won't go through all the methods here, but I wanted to show you that we've done some proteomics to look at the proteins which are contained within them. And we were hoping, obviously, to understand what proteins might be present in the exosomes which might mediate the protection. But when we looked at the, this is called a gene ontology analysis, where we can look at the main types of functions of the proteins that are identified in the exosomes, we found very high significance for all this vesicle-mediated transport and localization, which basically confirms the high quantity of exosomes in the solution. It doesn't give us much idea about how they might really be functioning. We did a targeted protein array to identify some signaling molecules which might be present, and we saw some proteins which we were interested in for various reasons, some which we'd investigated before, such as stromal-derived factor, and we thought these might be mediating some of the protection of the EVs. So we used inhibitors of these a few proteins of interest to investigate whether they might mediate protection. And to summarize, we didn't see any effect of the EVs, or the EVs in the presence of the inhibitors were similar. So we didn't see any specific effect of a particular protein. So we took a different approach. We reasoned that the exosomes might require a specific signaling pathway to activate signaling protection in the cells. And here we used an inhibitor of a PI3 kinase signaling pathway, which a lot of these proteins work on. And we saw a significant reduction in the effect of the exosomes when in the presence of these inhibitors. So to summarize, what we think is happening is that these exosomes contain a range of different proteins and potentially also microRNA, which I haven't spoken about today. I won't go into that unless there's some questions. But they might have a common signaling mechanism on the recipient cells to activate either the ERK signaling pathway or the PI3 kinase signaling pathway and protect the cells, the recipient cells. So to summarize then, it's important when working with exosomes to know about the limitations in their isolation, really. There are limitations of all different methods, and whether you're working with a pure or impure population of EVs, it's important to know exactly how pure they are and what they contain. Some of the signaling pathways mediated by exosomes from the blood are via this toll-like receptor and ERK signaling pathway. Importantly, diabetes can impair their function, but it's still potentially possible to use purified EVs as a means of protection, even in a setting of diabetes. And then as a potential therapy, obviously not very practical to isolate them from the blood all the time, but potentially we can isolate them from cultured stem cells or MSC cells. And these are an interesting source of potential protection or other signaling molecules in the cardiovascular system. So just, this is really an acknowledgement slide. This is where I work at University College London. This is our institute, And a lot of the work was done here in collaboration with Professor Yellen and by a PhD student and some collaborators as listed here. So I'll just stop there and happy to take any questions.
Thank you, Sean. Thank you very much. That was very interesting. I don't know if we have questions from the audience.
Hi, Cinzia. I'm Roberto Pedrinelli, and I would like to ask Sean two clarifications. The first is what's the difference in simple terms between exosomes and microparticles? Because it's unclear to me. Would you clarify this to me?
Sure. Yeah, thanks for the question. So these are similar, they have an overlap in terms of size, but because they're produced from cells by a different mechanism, they are believed to have different functions, and quite important different functions as well. Because if you remember the slide, the microvesicles are budding off the surface membrane of the cells, and therefore have a lot of surface membrane markers and receptors, and studies tend to indicate they're often pro-coagulant and detrimental in the cardiovascular system. Whereas exosomes are produced inside the cell, they don't have surface membrane receptors and released from the cells and are believed to signal then between different cell types, generally having beneficial effects, although I think that's yet to be fully confirmed.
Okay, and the second point that I would like to ask you is about the role of toll-like receptor 4, which in my view is usually associated with a pro-inflammatory reaction. But as far as I understand from your talk, they have a protective action, which usually is not mediated through inflammation.
Yeah, no, that's true. So I think it's a matter of dose. So with sepsis or massive infection, there is activation of the toll-like receptor TLR4 system, which is detrimental on the cardiovascular system. But it's been known for a while that if you knock out TLR4, then it's more difficult to induce cardioprotective strategies as well. So it seems to be part of a protection mechanism. If there's a small stimulus, you activate these pathways and you can get a protection, whereas a really strong stimulus can be damaging. And it's very similar to ischemia reperfusion. A small dose of ischemia reperfusion, it's like a preconditioning stimulus, which will protect the heart, but a long period of ischemia is damaging and reduces the injury. So I think that's what's happening here.
So this is a system which is operated through mechanisms which are either protective or detrimental, right?
I think an excessive stimulation of the TLR4, for example, is detrimental, but presumably it's not in the body to cause only detrimental effects. A small stimulation activates a protective signaling pathway and NF-kappa B as well, etc., which I haven't gone into.
Okay, thank you, and congratulations for your talk. This was very interesting.
Thank you.
Sure.
Sean, if I may, Massimiliano.
Yeah.
Two very outstanding presentation and lots of curiosity, but it's very easy for you. Biodistribution of extracellular vesicles, because as we know, once they are injected, they are cleared in minutes. So if you can comment on this aspect.
Yeah, I think it's still not well understood, I think, about the biodistribution of the EVs. And it's some things we're working on at the moment, but again, it's because it's difficult to isolate really pure EVs and to get good labeling. When you inject them, some studies indicate that they circulate for a while, but probably more convincing are the studies, as you say, which they are cleared quite rapidly. And in terms of targeting to the heart, I don't think many of them are taken up by the heart, especially prior to an injury. So with the heart, with its intact endothelial barrier, prevents access of the exosomes to the heart. But after an injury, such as ischemia reperfusion, when that's damaged, I think then these vesicles can reach the heart. But there's not a lot of evidence for that, and it's something we're trying to do now with the luciferase labeled EVs to see if we can get more uptake into the heart. I should say that other people, other investigators are administering them via direct injection into the myocardium for longer term procedures. But we found in several different types of EVs that we can administer them IV and get protection, possibly because they're accessing the heart following that injury.
Okay. And if I may, just another curiosity, because you have told that some of the effect mediated by extracellular vesicles are due to the content of protein and macroRNA. But do you believe that there is also a matter of lipids? So are they important, or in your opinion is something that we can not look at?
No, I think it's a great idea to look at that. I think it's been probably overlooked a bit about the lipids, but relative to their content, they have quite a high lipid content being so small. And I think there's definitely scope for signaling lipids, or even potentially oxidized lipids having a signaling mechanism, but it hasn't been, there are some people looking at it, but it's not greatly studied, I think.
Okay.
Definitely interesting question, yeah.
Thank you.
Yes, Sean, one quick question, actually two quick questions. Along the line of what Massimiliano just asked, has anyone ever looked at effects of lipid lowering agents or therapies on the amount and composition of exosomes or other vesicles?
Not that I know of offhand, I'd have to search for it, but yeah, I don't think so.
Because, you know, now there is a tendency to have the LDL levels, you know, the lowest, the better.
Yeah.
So I'm wondering whether this might, you know, affect the composition or the number of exosomes. And we already know. Another thing that always intrigued me is, why do you think all exosomes are protective? Are there exosomes that are detrimental? Because if they are, and you know, if the reason why they work in a certain fashion is because they contain something, there should be some beneficial and some detrimental, but both seem to be protective.
No, it's a good question. There are some examples of exosomes which are not protective. Mainly those from fibroblasts, for example, have been used. For some reason, I don't really understand why you can take exosomes from any type of stem cell and they seem to be beneficial, although there may be some differences in different sources of some might be better than others. But you're right, it's curious that they don't seem to have detrimental effects. Is it possible
that it's the machinery that makes them beneficial? So maybe the machinery that builds them up, and then when they get into the cells, it triggers something that is beneficial rather than their precise content.
Possibly, or I mean, I was encouraged by when we got our data that showed that the exosomes from the diabetic rats is not protective, because at least there's a difference there, and it shows it's not just an artifact of the isolation procedure that you take exosomes from anywhere and somehow for some reason you're causing them to become protective. At least there's some difference with the diabetic ones, which is, I think, interesting. But yeah, we still don't know exactly what is damaged in that setting of those exosomes.
Yeah, exactly. So if you don't find, you know, the mechanism, it's hard to understand why they all work.
Yeah, well, most of them work.
Yes.
Yeah.
Okay, I don't know if we have more questions.
Yes. I'm sorry, just a quick question in addition to the other one. Is there you mentioned that microparticles carry tissue factor. They are procoagulant particles, blood-borne particles, but is there any evidence for tissue factor in the exosomes, or can you exclude that tissue factor is involved in their action?
But I mean, most studies don't find tissue factor. I mean, there may be some which have, I don't know, but I'm not aware of any studies showing tissue factor in exosomes, because I think it's something which is found on the surface of membranes of cells typically. and platelets, for example, which then, when they release the microvesicles, they have the tissue factor on from the cell surface. I think that's the mechanism. So I believe the exosomes don't have tissue factor, and that's why they don't have the procoagulant effects.
Okay, thank you.
Are there any other questions?
Hi, I have a question. I'm Chiara from Milan. So congratulations for your talk. That was really an interesting talk, actually. With regard to the rat study, let's say, so the vesicles that you inject in the rat, did you sort the vesicles, or so did you know the origin of the vesicles that you inject in the rat, or are they just like isolated from blood, I suppose? So do you know if they are from macrophages, platelets, or whatever?
No, I mean, it's a good question. And we did this study quite a while ago when we were just getting interested in the field. So it was a bit crude, if you like. We just purified the exosomes that we could from the blood. And looking back now, it probably was not as pure as it could be. And we don't know specifically where the exosomes came from. Later on, we saw that there's a lot of platelet exosomes and endothelial exosomes. But it would be interesting now to take exosomes specifically from a platelet source or endothelial source or other source and see which ones are the protective ones. We don't really know that yet.
And so, okay, so I will add a question about that.
Yep.
So what do you think about sorting the vesicles? So do you think that it's harmful for the vesicles, or we actually can sort the vesicles and then, for example, decide to inject only the endothelial vesicles?
It's technically very challenging. Flow cytometry, you can't really do it. It's very difficult to use this, or you can't use a standard flow cytometer because they're too small, literally. There are some nano-sized nano flow cytometers, but they can't do the sorting yet. So you'd have to use some sort of affinity technique and then release them. So it's not very technically easy.
Yeah. Okay. Thank you.
Thank you.
So Sean, you got a lot of questions.
Great. Yeah, that's good.
That's really
Thank
good.
you.
That means it was a very interesting webinar. Thank you very much. I don't know if Massimiliano wants to add anything.
No, just really very interesting, and it has been demonstrated by lots of questions that you got.
So thank you again. Thank you also, Massimiliano, and thank you everyone for attending. So see, the basic science can be interesting also for a clinical cardiologist. So that's
we're trying to be translational.
Yes, we try
have
to
some patience.
Exactly. Yeah. Thank you very much. Thank you, everyone. And
thanks again for the invitation.
Good afternoon.
Good afternoon.
Bye-bye.
Bye.